Hi, this is the lecture for cellular anatomy. Let's get started. So, just for review, cells are the smallest living unit of life, and most are microscopic. Here are some examples of animal and plant cells. So, just to review again, Robert Hooke observed the first cells of cork, and they were dead, so he saw empty boxes, hence why he called them cells. Later on, the cell theory was invented. Theodor Schwann and Matthias Schleiden figured out that all living things are made of cells, and 50 years later, Rudolf Virchow figured out that all cells come from cells. So this led to our modern cell theory, and here they are in different words. You can pause here if you need to. So, the size of living things. Of course, an atom is one of the smaller things on our radar. Atoms, of course, are made of smaller parts. If you get a little bit bigger, you get to amino acids. Bigger than that is proteins. Bigger than that is viruses. Then, much bigger than that, you start to get parts of cells. Bacteria being unicellular, this being an organelle of a multicellular organism, and a human egg being a fairly big cell. This is all available to see on an electron microscope, and a light microscope sees a little bit less. And this is what we're able to see from the human eye. We can see all the way down to a human egg if you have good eyes. Okay. Then we talk about cell sizes. This is going the opposite direction. So this is the larger things over here going down smaller. So a plant cell is much bigger than a human blood cell, which is bigger still than a bacteria cell. And it's bigger than DNA and viruses. To review, I saw a few blank stares about the area to volume ratio. So imagine a cell was 2 centimeters by 2 centimeters. You'd have one cell in that space. If cells were 1 centimeter by 1 centimeter, the same space this one cell takes would have 8 cells in it. 1, 2, 3, 4, 5, 6, 7, 8. So if there were only one cell there, the total surface area would be 24 centimeters squared. If there were 8 cells in that same area, the total surface area would be 48 centimeters squared. Regardless of how many cells there are, the volume would remain the same. But if it were only one cell, the ratio of surface area to volume would be 3 to 1, whereas when there's 8 cells there, the ratio is 6 to 1. So there's a lot more surfaces for things to be going on, and a lot less space to travel for macromolecules and nutrients once in the cell. So some characteristics of all cells are they are surrounded by a membrane. They have a protoplasm, the cell contents in a thick fluid, which we call cytoplasm, and organelles, structures for cell function, and a control center with DNA. There's 2 different cell types, a prokaryotic cell and a eukaryotic cell. First, we'll talk about prokaryotic cells. In the 3 domains of life, bacteria and archaea are prokaryotic cells. Bacteria being the common things you've heard of, like E. coli and, well, none of these. Cyanobacteria is kind of algae. Are normal things that you see every day. Archaea are thermophiles. They like acidic things, basic things, cold things, hot things, or even salty. Halo means salty things. Salt lovers, halophiles. Thermo means temperature. And methano is methane. They like methane. So they like extreme conditions here. So both of these bacteria and archaea are prokaryotes, and eukarya, hence the name, are eukaryotes. All of these things here. Prokaryotic cells were the first type of cell on Earth according to evolutionary theory. They have no membrane-bound nucleus, so the DNA is just loose in a nucleoid region, and the organelles are not bound by membranes either. With the plasma membrane, prokaryotic cells typically have a cell wall and a capsule. The capsule surrounds both, and it says what it's made out of in the next slide. And it typically has a flagellum, some sort of thing for movement. Here's a visual summary of the prokaryotic cell. So, yes, the cellular envelopes. This is that capsule, glycocalyx is a layer of polysaccharides, so carbohydrates again, outside the cell wall, and it may be slimy and easily removed or well organized and resistant to removal. That is the capsule specifically. In prokaryotic cells, there is also a cytoplasm, and it has appendages, a flagella, normally for motility, and fimbria and sexpili. Sexpili is for sexual reproduction, for DNA to pass from cell to cell. It's not any more interesting than that. So eukaryotic cells are the domain eukarya. They include these types of things: protists, fungi, plants, and animals. The cells contain a membrane-bound nucleus, specialized organelles, and a plasma membrane. So here's the hypothesized origin of eukaryotic cells. This is evolution, and I'll get into the endosymbiotic theory later on in this. So a cell structure for eukaryotics is that they have first a cell membrane. The cell membrane is thin, it's dynamic, and it encloses the cell. The main model for it, since we can't clearly see it since it's so thin, is the fluid mosaic model. This is composed of a double layer of phospholipids and many protein molecules dispersed within it. So here's that lipid bilayer with the hydrophilic side out and the hydrophobic side in. There's also proteins embedded, either poking out on one side or going through the whole thing. They can have many different shapes. Here's another image of it that just shows how fluid the whole structure is. Think about it waving slightly as it moves. If you have forgotten, here's the lipid bilayer. If you need a little review, here's all of the information on lipids. So the plasma membrane serves as a boundary of the cell. It serves as markers that identify the cell. It plays a significant role in transportation, which we'll get into in the next section, and cell recognition, because some cells do recognize each other. And the membrane proteins, they can be channels or transporters, receptors, glycoproteins, or enzymes. You can read about this and pause if you feel like it. Cell walls are found in plants, fungi, and many protists. They surround the plasma membrane. In plants, they're mostly cellulose. In fungi, they contain chitin, which we've discussed a few times. So here's roughly what an animal cell can look like. Remember, that there are many different types of cell in an animal. And this is roughly what a plant cell can look like. Remember, there's also many different types of cells in a plant, different tissues. The cytoplasm is a gel-like matrix of water, enzymes, nutrients, waste, and gases, and it contains the cell structures, the organelles. The word matrix here just means a space where things can happen, a medium, and the medium here is the water with all these things in it. The fluid is called cytosol, and most of the cell's metabolic reactions occur in the cytoplasm. The cytoplasm is over 90% water. The organelles in the cell are machinery. So think about a car. All the different things in the car help the car run. So there's an engine, there's a steering wheel, there's windshield wipers, there's little mirrors on the side. So organelles are kind of like that. There's two general types, and there's those that are derived from membranes, so they're just membrane-bound organelles. And then there's bacteria-like organelles, which have a unique story, possibly. So the first and most important, if you're into genetics, the most important organelle is the nucleus. It is the command center, usually near the center. It has a nuclear envelope and a double layer of membrane. Nuclear pores permit exchange between nucleoplasm and cytoplasm. And it contains chromatin and semifluid nucleoplasm. The chromatin contains DNA of genes and also proteins, and it condenses to form chromosomes. We'll get more into that later on. And a dark nucleolus in the middle is composed of ribosomal RNA, rRNA. It produces subunits of ribosomes. Here is that nucleus. So here's the nucleus in a cell. Here's the actual picture of it. This is the outside surface of it. Up close are those pores. And the pores allow things in and out. There's the outer membrane and the inner membrane. And there's a bunch of space here with the nucleolus in the center with rRNA. The space in here has the chromatin. So ribosomes serve in protein synthesis, which we've seen a little bit. They're composed of rRNA, and they consist of a large subunit and small subunit. The subunits are made in the nucleolus. They may be located freely in the cytoplasm or in the ER. So the endomembrane system restricts enzymatic reaction to specific compartments within the cell. This consists of these organelles. Within the vesicles, several types are used, and they transport materials between organelles of the system. The endoplasmic reticulum, or the ER, is separated into two parts. The rough ER, which has ribosomes, which make it rough, and they're in charge of protein anabolism, which is the synthesis of proteins, and it also modifies proteins to add sugar to protein, creating glycoproteins. The smooth ER doesn't have ribosomes, and it's where the synthesis of lipids occurs. So here is that ER. It surrounds the nucleus, and the rough ER is in the center, the smooth ER is on the outer part. The Golgi apparatus, typically near the outside, consists of 3 to 20 flattened curved saccules and resembles stacks of hollow pancakes. It modifies proteins and lipids and packages them into vesicles. It receives vesicles from the ER on its face, and it prepares for shipment in vesicles from the transphase. So when it exports things from the cell, it's called secretion or exocytosis. We will get into that later as well. Lysosome are membrane-bound vesicles that aren't in plants, and they're produced by the Golgi apparatus. They have a low pH and contain lytic enzymes for digestion and cell death. Some genetic diseases are caused by a defect in the lysosomal enzyme, Tay-Sachs, for instance, which we will talk about later. So here's the endomembrane system at work. There's the nucleus with the rough ER and the smooth ER. Little vesicles pinch off and go to the Golgi apparatus, which sends little, this is the cis side closer to the inside, and then the trans is the opposite side, sends little vesicles to the outside of the cell, to the plasma membrane for secretion. It typically has things like sugar or proteins or lipids in these little vesicles. Vacuoles are membranous sacs that are larger than vesicles. They store materials that occur in excess. Others are very specialized, like the contractile vacuole. Plant cells typically have a central vacuole that is up to 90% of some of its cells, and it stores water and helps develop turgor pressure, which plants need to stay firm, and some functions performed by lysosomes in other eukaryotics. So here's that central vacuole in a plant cell. See how it takes up, in this cell, about half the space, maybe even 40%. Bacteria-like organelles are derived from symbiotic bacteria, possibly. This is evolutionary theory, and have an ancient association comprised through the endosymbiotic theory, which we'll talk about after we talk about the organelles. There's chloroplasts. They're bounded by a double membrane. The inner membrane is folded in and forms disc-like shapes called thylakoids, which are stacked into grana, and they're in a semifluid called stroma. The green color of these organelles is due to chlorophyll, which is a photosynthetic pigment, and they're found only in the inner membranes of chloroplasts, because that's where photosynthesis occurs. So the chloroplast captures light energy to drive cellular machinery. Photosynthesis synthesizes carbohydrates, which are what? Synthesizes sugars from CO2 and water. And it makes own food using CO2 as only carbon source. So here is a chloroplast structure. There's two membranes. The inner one is folded into a bunch of shapes called thylakoids. And that inner membrane is where photosynthesis occurs. Mitochondria. They're also bounded by a double membrane. That's an important thing to remember. The cristae are the enfoldings of the inner membrane that encloses the matrix. And the matrix is an inner semifluid containing respiratory enzymes. This organelle is involved in cellular respiration, and it produces most of ATP, which is an energy unit utilized by the cell. Here it is. Here's that double membrane, and the matrix is the space in between the cristae, which are the folds. So endosymbiotic theory is the theory that both mitochondria and a plastid like chloroplast were once individual cells that had a symbiotic sort of relationship with these eukaryotic cells. So bacteria cells that had a relationship with these bigger cells. And it's thought they had such a close relationship that through vaginosis, where the membrane pinched it off and made it a vesicle, these organelles became part of the cell. Note it, as I said earlier, they both have two membranes. So the mitochondria has two membranes, and also the chloroplast has two membranes. And this is thought that it's their original bacterial membrane surrounded by the plasma membrane of the cells. One reason people believe this is true is because mitochondria has its own DNA. And why would something have its own DNA? It doesn't make much sense if why would something have its own DNA? It's a mystery. So there's no if about it. It's confusing. You get mitochondria only from your mother too. It's most everything else in your body comes from both your mother and your father, but the mitochondrial DNA is only from your mother. So if you look at mitochondria, that is a direct line back from every woman who was ever a mother in your family. So your mother, your mother's mother, the mother of your mother's mother. Anyway, your maternal line. That brings us on to the cytoskeleton. It maintains the cell's shape and assists in movement of cell and organelles. There are three types of macromolecular fibers: actin filaments, intermediate filaments, and microtubules. They assemble and disassemble as needed. So actin filaments are extremely thin filaments, like twisted pearl necklaces. There's a dense web just under the plasma membrane that maintains cell shape. You could see it in one of the first slides. And it's support for microvilli and intestinal cells. They also help move things around. We won't talk about the pseudopods or amoeboids necessarily, but they pinch the mother cell in two after animal mitosis. So when a cell goes through mitosis, which we'll go into in more detail, there's two cells forming kind of in a long shape, and there's the actin filaments pinching them apart, like a rubber band almost. And they are an important component in muscle contraction. So here's an actin filament. See these little tiny fibers going around each other. And that is to do with muscle contraction. Intermediate filaments is the size between actin filaments and microtubules. They're a rope-like assembly of fibrous polypeptides. They vary in nature from tissue to tissue and time to time. They support the nuclear envelope, so around the nucleus, and they're part of cell-to-cell junctions. Microtubules are a hollow cylinder made of two globular proteins called alpha and beta tubulin. Spontaneous pairing of alpha and beta tubulin molecules form structures called dimers. Dimers then arrange themselves into tubular spirals of 13 dimers around. You don't need to worry about this stuff. So here they are. There's the alpha and beta wrapping up, creating dimers. And so this whole structure is the microtubule. Microtubules are cool because vesicles, little tiny things going out, taking lipids and proteins and sugars outside of the cell or into the cell. These vesicles move along. So these two little things are like feet, and they kind of just walk along until they reach the end of the cell and to release things. We'll watch a video on it. It's pretty neat. So here's those three in action. Actin, you can barely see them here. They're kind of just little tiny structures. Intermediate filaments, you can now see them. They clearly show lines here. And then the microtubules are much thicker. So, microtubular arrays include centrioles, which are short, hollow cylinders composed of 27 microtubules. And they're located in centrosome of animal cells. They help with cell division. So when the DNA divides and two daughter cells are about to form, there's a wall here, these centrioles come in between and pull the DNA to opposite ends. So these were all together in the middle, and now they're being separated, being pulled to either side. There are also little modifications on the surface of the cell membrane. Cilia or cilium, which are short hair-like extensions, kind of like eyelashes, and they help things move through passageways. They're located on the lining of the respiratory tract and the fallopian tubes of that go to the eggs and the ovaries. And the flagella is one long structure like seen on sperm. They're tail-like projections, and there's only one per cell in humans, and they aid in locomotion. Microvilli are small finger-like projections, and they increase surface area. They help absorb food particles in the digestive tract. Membrane junctions. There's also tight junction, desmosomes, and gap junctions. A tight junction is an impermeable. That means you cannot permeate it, you can't go through it. That encircles the cell and prevents leakage. This is important in skin. Like, you don't want anything going through your skin, right? Because you touch toxic things all the time. And if everything you touched went through your skin, we'd be dead. Also, blood-brain barrier. You don't want things just going into your brain willy-nilly. Desmosome are anchoring junctions scattered along the side, and they prevent tissues from fraying. So they're kind of like stitching. They're in the stomach and the uterus and the bladder. And gap junction allows chemical substances to pass between cells. This is common in the heart, where you want blood to flow, right? Here they are. Here's a tight junction. Interlocking junctional proteins. The proteins hold them closely together. Then there's the desmosome, which is like stitching. Okay? Linker proteins. Again, proteins can do about anything. They can look about anyway. Stitch them sort of together. And there's a small intercellular space. And the gap junction. Again, proteins that make little channels in between the cells and give a larger space for things to go between cells. So here's a review. I'll pause on each slide for a second. This is the outside of a cell. These are different types of organelles inside the cell, their description and their function. I'll move that. And the differences between bacterial, animal, and plant cells. All right, have a great day.